Hello, everyone. My name is Chris Leptak, and I'm the director of CDER's Biomarker Qualification program at the FDA. I appreciate the workshop organizers inviting me to give this talk today around how we view biomarkers from a regulatory perspective. And so in the next 15 minutes or so, I'm going to give you a high-level presentation of the types of biomarkers, the roles in drug development, and then also to interplay that with a little bit of how biomarkers may be used in the pain setting. I do need to acknowledge up front that for the most part, since pain is an assessment of how a patient feels or functions, there's an opportunity to ask the patient directly. So the use of biomarkers in the setting of pain treatment has not, at least historically, been a very common goal, but nonetheless, I know that there is some interest in this. So I will be covering the details of biomarker development from a regulatory perspective. As with anyone at the FDA, I don't have any information to disclose. If I did, just couldn't have my job. So that was always kind of easy. So ultimately, let's start with fundamentals, and that's the definition of a biomarker. And as it's defined, it's a defined characteristic that is measured as an indicator of normal or pathologic biological process for response to an intervention. When we think of biomarkers, we think of them very broadly. I think in terms of the lay press and in the media, a lot of times people equivocate biomarkers to something that's in a bodily fluid. But it can also be a radiographic finding where you're looking at anatomic structures, or from a biopsy looking at histologic features, or even a physiologic output for an organ system. But what a biomarker is not is how a patient feels, functions, and survives. That's a clinical assessment, and it's contrasted with what we call clinical outcome assessments, or COAs, at the FDA. Although my presentation today is talking about biomarkers and the process for development known as qualification, much of the material around the process itself would also apply to COAs. Ultimately, there's many different communities that do use biomarkers. They're not exclusive to just regulatory use. And so certainly clinical folks use them as part of patient management. And in addition, academic folks who are publishing in the scientific journals are ultimately communicating around biomarkers from basic science perspective. Ultimately, we're asked, if one of these communities is accepting of a biomarker, why can't all of the communities? And ultimately, from a regulatory point of view, some of the concerns around our use of biomarkers that are either out in the publications or that are even used in clinical practice, there are some limitations. One is the reproducibility of the data, and that there's a high rate of discordance between conclusions in biomarker published reports. When you're looking at a meta-analysis of all the reports on the same biomarker for the same disease or use, and many times there's a fair bit of disagreement around the overall utility. Also, since the biomarkers ultimately be measured, the analytic device for measurement, many times there's a problem with the adequacy of the biomarkers' analytical performance. And as well as the published literature, many times many different platforms or measurement methods are used, and there's not enough detail around the measurement methods and how they were employed for us to be able to pool information to do a more meta-analysis between studies. And finally, if you're going to use the biomarker for regulatory purposes, in our case, drug development, then that biomarker needs to be practically available out in the community, so that if clinicians want to use that biomarker as part of their own decision making, they need to have access to the tools that it was used in the clinical trial setting. So while it is possible that biomarkers can be used by many different communities, there are some unique differences between those communities. I would like to draw your attention to a resource that's been out now for a number of years. It's referred to as BEST, which is the Biomarkers, Endpoints, and Other Tools resource. It's a glossary of terminology in the biomarker space, and it's publicly hosted on the National Library of Medicine website. The link is here. It is a living resource in that it's periodically updated. And in fact, the working group between NIH and FDA are working now based on some FR notice comments, making some additional updates to the resource. Although the BEST covers many different topic areas, from a biomarkers perspective, it does define a series of classes that ultimately is determined by the use of the biomarker in a drug development or clinical trial context. Ultimately, the group came up with 7 different classes of biomarkers that are listed here. I'm not going to go through and have the definitions. I think it's more important to kind of think about them from a sort of practical perspective. And so on this next slide, you'll see that there's putting those best classes of biomarkers into a fit-for-purpose diagram. So if you start in the top left, which is described as normal physiology, ultimately, this is the setting of what is the normal variation of a biomarker as part of its biologic function. And so, for instance, for a given person throughout a 24-hour period, depending on what the biomarker of interest is, that biomarker may change based on either normal cellular function or organ function. Likewise, there may be changes from day to day. There may be changes between individuals. And those changes may be reflective of either age or sex or other demographic factors. So as part of a biomarker development effort, it's really important to know what your normal variation is in an unperturbed setting, so that if you do focus on that biomarker down the road, you would know what change of variation is above and beyond what would normally be expected. In this normal physiology state, ultimately, we can be talking about susceptibility risk biomarkers, where ultimately you're trying to predict the development of disease where disease currently does not exist. And so that would be the one class. As organic beings, certainly there's a change or an evolution over time of how our cells and bodily function. And those changes can result in some cellular changes that ultimately can result in altered physiology. And then if that physiology gets so altered that you get signs and symptoms that we then define as disease, you then get into the next set of best characteristics referred to as diagnostic, monitoring, or prognostic. Diagnostic falls in 2 flavors. One is that the biomarker in and of itself is necessary and sufficient for diagnosis of the disease, or it could be a lowercase d diagnostic, and that, in fact, in order to diagnose the disease, you need multiple different inputs for the diagnosis, some of which may be biomarker, which would include laboratory or imaging studies. It could also be a constellation of signs and symptoms. In addition, once you have disease, sometimes for some of our chronic conditions, they can wax and wane in their activity level. And then we get into a class of biomarkers known as monitoring. So basically, you can do serial assessments over time to see what the level of activity of that disease is, as an example. And then finally, the third best category here is prognostic. And that's saying that once you have a disease, what's the likelihood that you're going to develop a clinical event of interest in the future? An example would be in the setting of COPD, what's the likelihood that you're going to have a hospitalization or a COPD exacerbation within a certain time frame? All of these biomarkers that classes that I've mentioned so far are all without respect to any type of intervention or treatment. Once you do have a therapy in play, you then get into the remaining classes of biomarkers, and that ultimately, if you do have disease and you're treating that individual, you ultimately are hoping that there's going to be a change in the physiology. And that physiologic change may, in fact, result in either a slowing of the condition or that, in fact, it ceases progression or maybe even improves. So the first class of biomarker here is the pharmacodynamic class. And here we're talking about that there's an engagement between the therapy and some cellular or molecular function. It doesn't say that the outcome of that engagement is either beneficial or detrimental. It just says that an interaction has occurred. So if you think of a biologic therapy, if it's engaging with the cell surface receptor, that interaction could be assessed as part of a pharmacodynamic response. Again, it doesn't suggest any type of efficacy or benefit. Another class of biomarker here is predictive, and here's where you're trying to take patients with a known disease and subset them around the likelihood that they'll either have a beneficial response to the therapy or potentially have a higher likelihood of an adverse outcome. And then finally, there's the safety biomarker, which are looking for organ toxicity effects that are off target or to be not the expected pathway for the therapeutic intervention. If that change in physiology ultimately results in non-reversal or non-progression, we can get into a response biomarker category, and then ultimately, if that response is either reasonably likely or has been shown to predict clinical benefit, then we get into the surrogate endpoint class. When you're talking about a biomarker's use in a clinical trial context, you want to know what the value or utility of that biomarker is, and we refer to that as the context of use. Simply speaking, the context of use is the best biomarker category, one of those 7 that we just described, and then how that biomarker impacts the clinical trial or drug development program. Those uses can be many. For instance, they can be involved as part of inclusion-exclusion criteria to define the patient population of interest. It could be used to design the trial to see whether or not there's a safety concern that perhaps you can monitor or mitigate. And then there's also the interest in using these as endpoints to determine whether or not the therapy is efficacious in that given patient population. Biomarkers can come into the agency through several different pathways. There's the drug approval process, which is the most long-standing and still the highest, the pathway with still the highest level of interest. And that's where individual sponsors of INDs are bringing forward biomarkers as part of their individual drug program. Sometimes these biomarkers are ones that are already accepted by the community at large. Sometimes they're novel in nature. There's also scientific community consensus, which is ultimately taking information from the published literature or from professional society recommendations. And ultimately, here in the scientific community consensus is that there's a growing body of evidence that accumulates over time. So the biomarker has a particular use for a given patient, and that use is defined. And then finally, there's the Biomarker Qualification Program, which is the development of a biomarker independent of a specific drug program. All 3 of these, it should be noted, use the same terminology and are evaluated by the agency in the same way, in the sense that there are teams that includes representatives from many of the components of CEDAR, even sometimes other centers, that bring forward their discipline-specific expertise to the biomarker development consideration. That could be from statistics, from clinical pharmacology, from pharmacology, toxicology, et cetera. So they all share core concepts. They're all data-driven. And honestly, at the FDA, we don't have a preference for how biomarker information comes to us. We certainly will consider it through any of the pathways. And many times for novel biomarker development, information is brought forward from more than one pathway. The FDA has been qualifying drug development tools through a guidance-driven process that started in 2006, 2007, and that got formalized as part of the 21st Century Cures legislation back in December of 2016. At the same time, we were undergoing a PDUFA 6 reauthorization, and so a number of biomarker-related topics were brought forward. So one of the benefits of the Cures legislation, as well as the PDUFA 6 negotiation, is that it increasingly placed FDA as an active participant in drug development, specifically around the development of drug development tools, or DDTs. They also formalized a 3-step submission process for the development of DDTs, namely the letter of intent, the qualification plan, and the full qualification package. The letter of intent is relatively short, and what you're trying to bring forward at that point is an overview of what the drug development tool is, what the drug development need is that the tool is intended to address, and some summaries of preliminary data to support your hypothesis. The letter of intent is not a data evaluation on our part, so you should be encouraged to engage with us early on in your project development so that we can give you feedback around that development, which will make your time and resources more efficient. The second step in the qualification is the qualification plan, and here's where you really provide a high level of scientific detail around your overall development effort. You're summarizing the existing data as it is at the time of creation of the QP, how you're interpreting that data, what your overall goal is, what scientific gaps may be in your way to get to that goal, and how you plan to fill them or mitigate them. And then finally, the full qualification package, or FQP, is the final submission where you provide the data, your analysis, and your interpretation, and it gives us an opportunity to look at that same data to see if it comes to the same conclusion. One of the real benefits of the 507 process, which is the 21st Century Cures process, is that it's transparent. If you go onto the qualification program web pages for either biomarker COAs or for animal models, you'll find a listing of the projects that are currently in our programs, as well as information around the submissions that we received and our responses to them, including the recommendations that we have for further development. So this is a real opportunity for a shared learning culture, in that if you're developing a biomarker for a disease or using a certain measurement technology, you can see what other individuals are doing in that same space, look at the recommendations we received, and incorporate those as part of your overall development plan. What we found in even the few years that we've been following the 507 process is that the quality of the applications has really improved dramatically, which has enabled people to get to their goals more quickly. And then finally, as part of the Cures legislation, we have put forward in guidance reasonable timeframes for the review of the 3 different submission types. There is a listing of, like I said, of the projects that are under development or those that have been qualified. And I have this slide here more for reference. These slides are available, and you can follow the link. The same would be true for the COA program. And then I'd like to end with just 2 slides in the setting of surrogates. Certainly, that's surrogate endpoints are of broad interest to both drug developers as well as the patient groups. And so as part of the Cures legislation, we were required to bring forward a list of curated surrogate endpoints that can be used for drug approval, whether accelerated or traditional. That's been now publicly available for about 2 years. We update it every 6 months. And ultimately, I think what you'll find if you do look at that table is that whereas many people think that the FDA only uses a handful of surrogates, that what comes to mind are HIV viral load or LDL cholesterol or blood glucose. But in fact, we use surrogates for many, many diseases. And as you'll see on the tables as they're presented, there's over 100 adult conditions and 56 pediatric conditions for which the surrogates have been and continue to be used on a regular basis. And that many of these drug approvals ultimately can lead to faster drug development. Another surrogate resource that's available is if you're a drug developer and you have a novel surrogate of interest, you can request what's called an IND Type C meeting for the discussion of that novel surrogate. The web page link that's provided here brings to your attention a content outline for your meeting briefing document that gives you a skeleton to follow of how to craft your proposal in a way that brings forward your arguments hopefully persuasively. So for instance, the package includes a rationale for the surrogate endpoint. In that case, we would say, if there's a hard clinical endpoint of feels, functions, and survives, why do you need to use the surrogate? Because if ultimately you're going to introduce uncertainty, which surrogates do, and a hard clinical endpoint is available to you, we would certainly encourage you to continue to use the direct measurement of how a patient benefits. There's also additional topic areas to be covered, including how the surrogate endpoint relates to the causal pathway for the disease, the threshold of change to demonstrate clinical relevance, the consistency of the surrogate endpoint response across the population that's being studied, et cetera. So the bottom line is, although we do use surrogates, and especially in the setting of trying to mitigate pain in patient populations, for the most part, certainly historically, it's better to assess from a patient directly how they're responding to their pain. If you are going to bring forward a biomarker proposal, do kind of keep in mind, again, you have to make the argument of why a biomarker could be beneficial in this case. And with that, I would like to thank you for your attention. And I understand from the conference that there's going to be a live panel discussion to supplement these recorded presentations. Thank you for your time.